ADHD is a mental disorder characterized by attention difficulty and impulsive behaviors. The precise causes for ADHD vary and are not really known. However, the disorder is linked with lower levels of monoamine neurotransmitters in neural synapses. These neurotransmitters, particularly dopamine and norepinephrine, regulate important cognitive processes like motivational salience and mental alertness. Amphetamine, commonly known by the brand name Adderall, is one of the most common treatments for ADHD, and it works by boosting the levels of monoamine neurotransmitters such as dopamine in the neural synapses. Amphetamine enters the neuron through dopamine transporter proteins. These carrier proteins also allow the influx of dopamine. Once inside the cell, amphetamine completes two functions. Firstly, it acts as a competitive inhibitor to the uptake of dopamine through vesicular monoamine transporter 2 proteins. This causes the concentration of dopamine in the cytosol to increase. The second function has a longer pathway. It begins when amphetamine binds to a trace amine associated receptor 1 protein, which is coupled to the G proteins GQ and GS. The GQ protein activates the enzyme phospholipase C, which catalyzes the conversion of PIP2 to the second messenger diaglycerol. Through a very similar process, the GS protein produces another second messenger, CAMP. CAMP activates the protein kinase PKA, diaglycerol activates the protein kinase PKC. The protein kinases finally phosphorylate DAT. If PK phosphorylates DAT, then the transport protein internalizes and it becomes unable to let dopamine from the synaptic cleft back into the cell. If PKC phosphorylates DAT, then the transport protein reverses function to allow dopamine efflux instead of influx. The inhibition of VMAT2 and phosphorylation of DAT proteins facilitate dopamine efflux and prevent dopamine influx. This results ultimately in a higher concentration of dopamine in the synaptic cleft where it can bind to dopamine receptors.